Can you hear me okay? There you go. All right. Well, look, I'm really grateful for the opportunity to speak to you. Honored, actually. I'm excited about the fact that so many people here seem to have an interest in this class of lipids that I've been working on for a little while now. And I'm aware of the fact that I'm one of the last things standing between you and dinner. So let's get right to it. So, first I need to disclose that I'm going to show you some data from a biotechnology company that I helped form a few years ago, and so it has a chance to make me money, so you can disregard what I'm going to say as you wish. So my lab is his work is predicated on the idea that diabetes and heart disease really result from too little fat. They result from the inability of our fat stores to safely store the nutrients that we bring in. And that's either because we maybe eat too much, like I do, or perhaps there's some kind of defect in the adipose depot that prevents them from being stored safely as triglyceride. And when that happens, when there's an impairment in that adipose depot, then the fat starts to spill out into places where it doesn't belong, like the heart, like blood vessels, like the liver, like the pancreas. And this is what gives rise to these cardiometabolic disorders associated with obesity and dyslipidemia. So a number of years ago, we started trying to map which fats actually drove tissue dysfunction. And we started doing this in cells by just dumping on huge amounts of fat. Now, when fat gets to a tissue, it really has 3 different metabolic fates. So it's immediately converted to acyl-CoAs. But it can then be coupled to carnitine, where it's shunted into mitochondria to be used to produce energy. It can be coupled to glycerol, which allows it to enter the glycerolipid pathway, which allows it to be converted to triglycerides and stored for subsequent use. And there's a clear evolutionary basis for each of those. The organism needs to produce energy, and it needs to store energy for times when nutrients are scarce. A little bit can spill over into this third pathway, be coupled to serine, and produce sphingolipids, including the ceramides we've been hearing about. And I want to talk a little bit about what those do and what the evolutionary basis might be for this pathway existing. Now, first of all, this is a pretty rare class of lipids. It's much less abundant than the glycerolipid pathway. Probably 1/17 as much goes into that pathway in a normal cell. And if you see here, this is the actual amount of ceramide that's present in this particular tissue lipidome that we measured. So the ceramides are a relatively small component. What I'm going to try and convince you of today is that this pathway evolved. Our cells evolved to use this pathway as a way of preventing fatty acid toxicity, and that they initiate a series of cellular actions that prevent the cell membrane from just lysing under conditions of these detergent-like fatty acids accumulating, getting too high, and actually lysing the cell membrane. So they form detergent-resistant membranes. They do a series of actions to lower cellular fatty acid levels. And lastly, they inhibit glucose utilization, which is what then leads the cell to preferentially burn fat as its preferred energy source. But if you think about what that is in terms of acute fatty acid toxicity, those are all good things for a cell. But in the case of obesity, chronic impairment in glucose utilization and chronic upregulation in lipid storage is selective insulin resistance. It is prediabetes. Now, what happens when this pathway continues to get filled up, when the energetic needs have been saturated and the storage pathway has been saturated, fatty acids continue to spill into this pathway to the point that the cell has no option other than to kill itself for the sake of the organism and to induce fibrosis for the sake of the organism. And we think these are the transitions that actually take you from this pre-diabetic condition into things like heart failure. Now, we've been working on ceramides for about 20 years, and there's been quite an increase in interest, and I think what's happening in Finland right now is going to be a big reason why this curve will continue to go up. the types of articles that have been done looking at the relationship between ceramides and metabolic disease fall into one of four categories. So first of all, they started off in the late 90s where we just started dumping ceramides, our fatty acids, on cells and asking, what did these ceramides do? And we discovered some of the things that I'm going to tell you about later, that they impaired insulin action, for example, and induced insulin resistance. Subsequent to that, there were a series of interventional studies that were done in the early 2000s, later, towards 2010, where people started manipulating ceramides in rodents and looking at cardiometabolic endpoints, and I'll tell you a little bit about those too. More recently, with the ability to do larger scale lipidomics and with increased ability to do genetics, we've started to see relationships in humans between the ceramide profiles, as you've heard about so much, and genetic mutations in the ceramide synthesizing genes that also seem to influence one's susceptibility to cardiometabolic disease. So just briefly to share the summary of those as I see them. The early studies where people would either dump on ceramide analogs or they would dump on the precursors of ceramide and then manipulate the endogenous pathway showed clear roles for ceramide in a whole series of actions relevant to diabetes and heart disease. So in skeletal muscle and adipose tissue, for example, it induces insulin resistance. In the liver, it induces insulin resistance and seems to drive triglyceride deposition and induce steatosis. In the heart and vasculature, it decreases vascular reactivity, seems to contribute to cardiomyopathy and perhaps cardiomyocyte apoptosis. And you just heard some stuff about how in the serum it also contributes to aggregation of these lipoprotein particles, which is also quite interesting. In the pancreas, it actually decreases insulin gene transcription and induces death. So all of these things are relevant to diabetes, diabetic complications, and cardiovascular disease that is, of course, the leading cause of death for diabetes. So one of the early studies we did in rodents when we started to do interventions was we manipulated this pathway by modulating one of the four biosynthetic enzymes that links exogenous palmitate, serine, to the formation of ceramide. And one of the best compounds we had was this drug, myriocin. Now, myriocin is actually has been isolated from a fungus that grows out of dying caterpillars in Tibet, called myriocin. And this traditional Chinese medicine is used for a lot of things, including diabetes and heart disease. And it turns out the concentration of myriocin in that varies widely by where you get it from. Get the stuff from Tibet that's expensive, not the stuff that's cultivated. But it's actually been used, for a long time. But we took the compound myriocin, gave it to rats and mice, and it's also been given to hamsters. This happens to be a rat study that we did years ago, where we just started treating with myriocin every other day. This is a leptin receptor deficient rat. It's super fat. It eats insatiably. It develops hyperglycemia and hyperlipidemia. And what you see here is if you start treating the animal when it's 8 weeks of age, then within 4 weeks or so, the normal vehicle-treated rat becomes hyperglycemic and it becomes hypertriglyceridemic. So it's got high glucose and high lipid in the blood. If we treat with myriocin, it stays fat. It's just as fat as it was before, but it never develops diabetes. It never develops insulin resistance. It never develops the hyperlipidemia. So we protect from those two disorders. That has been done and repeated with a lot of approaches. I'm going to show you some more data when I get a little bit later. This has now been shown, that myriocin compound has been shown to prevent atherosclerosis. It's been shown to prevent fatty liver disease in mice. It's been shown to prevent type 2 diabetes. It's actually been shown to prevent type 1 diabetes in mice. And it's been shown to prevent hypertension. So based on those observations, we became quite excited about ceramides and decided to commit some effort to understanding why they were so toxic. Now, in terms of human studies, there's been a little bit of controversy. And the early controversy was because some small, in my opinion, underpowered studies were reporting no associations between ceramides and insulin resistance. And this led many people in the field to decide ceramides, there was nothing to see here, and they should go in a different direction. But of course, with what's happened recently with the larger scale lipidomics, you're seeing these associations that we've heard so much about between ceramides and coronary artery disease, ceramides and insulin resistance, and other metabolic indices. We've jumped on the bandwagon. We profiled a Utah cohort of about 700 patients. We saw the same type of thing that's being reported by so much more impressively in Finland, where certain ceramides seem to associate strongly with the disease progression, and these are independent of a whole series of risk factors. You've heard about this today, and you've heard about it much better than we could do it. But the same thing seems to happen in Utahns as opposed to other cohorts. This has been done by a lot of different groups. I think the best work is the work we've been hearing about by Reiska and his colleagues. The Strong Heart Family Study has recently published a couple of very large studies showing relationships between ceramides and insulin resistance in Native American populations. And so I think this is continuing to get stronger and stronger and showing these relationships between a whole host of different disease endpoints. And of course, it's this that has given rise to the test that Zora has been marketing, and the test that's available through the Mayo Clinic. That's the image from their website where they're marketing this. I first learned about this when I started having people call me and say, my ceramides are high, what do I do? And since I work on mice, I had a limited ability to give any kind of really good answers to that, but I actually think it's a real problem. I think the fact that we don't have really clear indications, we're testing something in people without a clear evidence of what's the best approach to actually treat them and lower their ceramides. So I think we really do need to get on the ball and get there. So what I'm going to do is I'm going to walk you through the 5 W's of sphingolipids and metabolic disease. So the who, what, where, when, and why. And this has been the framework for what we do in the lab for a long, long time. The first question I want to address is who. And what I mean by that is this pathway is complicated. And if you haven't picked up on it, the nomenclature is kind of brutal. So the complexity, there are about 4,000 different sphingolipids that have now been identified downstream of this pathway. The complexity results from 2 different modifications. First of all, to the sphingosine backbone, which is created from this condensation of palmitate and serine, a second acyl chain can get added. And this gets added by a family of ceramide synthase enzymes. They can add long-chain ceramides, which are like C16s that we've heard about, and those are the ones we think are really bad. Or they can add very long-chain ceramides, 24, 24-1, or even longer in the skin and in the retina. There are short-chain ceramides, but they're not very biologically relevant. They're actually used more as tools. The other complexity comes because the head groups, you can add different head groups to that ceramide scaffold. And this is actually where the more abundant sphingolipids come from. So the most abundant is sphingomyelin, which has a choline group added to it. It can also be phosphorylated. Ceramide can also have sugar residues added, and that's what gives rise to gangliosides. Or it can be deacylated to produce sphingosine and another product, sphingosine 1-phosphate. So we've been playing around with this, and we played with all these enzymes that add head groups. None of them made anything better. They all made something worse in our hands. There's a little bit of evidence for gangliosides doing something relevant to atherosclerosis and metabolic disease, but what they're doing and how they're doing it is a little more controversial. We're quite convinced that ceramides themselves turned out to be one of the harmful sphingolipids that's produced. And in fact, our work has led us to believe that C16 ceramides are particularly damaging. And 16s are interesting because that fatty acid chain has the same number of carbons as the sphingosine backbone. And it seems to partition in membranes a little differently than those that have the really long chain acyl chains added to it. So these acyl chains can be added by 6 different ceramide synthases. They differ in substrate specificity, so they'll add different acyl chains. They differ in tissue distribution. We decided a number of years ago that we would go after this one that's in red. This is ceramide synthase 2. This is the dominant isoform in the liver. It makes the really long chain ceramides. We thought we're going to show that this is effective, we're going to make a drug against it, and I'm going to get rich. So that was the grand plan. That did not work, so I am not rich. And in fact, we not only didn't make things better, we made things worse. So what happened is we took a mouse that was heterozygous for ceramide synthase 2, and sure enough, we showed that we could decrease the production of these very long chain ceramides that were the product of ceramide synthase 2. But what happened was there was a compensatory increase in the ceramide synthases that made the C16 ceramides. So we ended up with about the same number of ceramides, but we ended up with a change in the ratio so that we had a lot more of these C16s and a lot fewer of these C24s. And when you put on a chow diet, these animals were indistinguishable. They looked completely healthy and completely fine. We put them on our standard obesogenic high-fat diet regimen, and the animal got much, much worse and developed full-blown NASH very, very quickly. Insulin resistance, hyperinsulinemia. Here's a little bit of the data showing that on chow, both the wild-type animals and the heterozygous animals had comparable insulin. They had no lipid deposition in the liver. If you put them on a high-fat diet, the wild-type animals started to show insulin resistance as measured by hyperinsulinemia in this case. We did a bunch of other things too. The heterozygotes for the SIRS2 were much, much worse, accumulated a lot more of the C16 ceramides, and they also accumulated a lot more fat in the liver. We later went on to show that it was the C16 ceramides, that it was SIRS6. We went back and added viruses, proved that it was the ceramide synthase 6 that was doing that. And simultaneously, Jens Bruning's group found that ceramide synthase 6 was the driving factor for those C16 ceramides. He got rid of ceramide synthase 6. It did make everything better, and he got involved with a company to start trying to make inhibitors against ceramide synthase 6. So that was really good validation and convinced us that these ceramide synthases, that ceramide synthase 6 was particularly bad. Ceramide synthase 5 also makes C16 ceramides and seems to have a role in damage to the heart. So there seem to be some other ceramide synthases that may be good drug targets in that there as well. Ceramide synthase 2 is clearly not. As a matter of fact, there's a mutation, there's a very common SNP in Northern Europeans. Sorry. 1 in 4 people, or 1 in 5 people of Northern European descent, about 19% of Finnish people have a mutation in ceramide synthase 2. We think this is an inactivating mutation, and people with this variant, one copy of this variant, have increased HbA1c and increased diabetic kidney disease. And we're trying to do the profiles now to relate the SNP to the ceramide profiles. And those are just the numbers in the population, about 20%. It's quite variable. It's actually very low in Asians, for example. Now, just briefly, I want to tell you about one exciting development that's new, and then I'm going to get to some new and exciting unpublished stuff, or at least I think it's exciting. And that's this issue of when. And what I mean by that is, when are these ceramides getting turned on? Why would it happen that they go up in some people and not others? And what are the factors that regulate the flow of carbons and the flow of fat through that biosynthetic pathway? And we now know that they're influenced by saturated fats predominantly. They preferentially go into that pathway. Palmitate's required for induction of that, or for that creation of that sphingosine backbone. We know that they're induced by just about every inflammatory cytokine. One of the things they do is they turn on this pathway. They upregulate a series of the genes in the pathway. They do so through NF-kappa B. We've mapped that pathway. Glucocorticoids also turn this on. This is a very generic stress response that seems to get turned on by a lot of things. We know that they go down with cold. We know that they go down with exercise. I'm going to show you the beta-adrenergic stuff in a little bit. That's new. One of the more exciting developments was adiponectin. So some of you may have heard of adiponectin. It's adipokine that's secreted from adipose tissue. It's anti-diabetic. It's cardioprotective. It's secreted when adipocytes are small and healthy. When they get big and unhealthy, then the body starts to produce less adiponectin. The adiponectin receptor looks like a ceramidase, an enzyme that degrades ceramides. So Will Holland and Philip Scheer went in, and Will runs a joint lab with me. They went in and mutated the residues in that ceramidase domain and found out that adiponectin was then inactive. If they expressed into ceramidase, they could recapitulate all the things that adiponectin did. So it looked like adiponectin elicited these anti-diabetic and cardioprotective actions by degrading ceramides. They showed that it could in fact do that. And then shortly thereafter, Sebastian Granier solved the crystal structure. This was in Nature last year, showing that it was a ceramidase, it had ceramidase activity. Structurally, it looks just like a ceramidase. So this was a big development for us because it showed a new way that this pathway was regulated and that ceramide levels were modulated in people. There have been clinical studies now that are starting to come out correlating adiponectin with ceramide levels and showing this inverse relationship. Okay, so now I want to get to sort of the things that really bother us as a laboratory and that we spend a lot of time thinking about. And that is, what do these ceramides do? Where do they do it? What tissue is, where is the target tissue of interest? Where in the cell are they acting? And lastly, this idea of why the pathway evolved in the way that I've tried to describe it as this means of protecting the cell from fatty acid excess. So our way of doing this has been to go to the mouse and to manipulate the enzymes that produce ceramide in a way that we can either reduce the ceramide levels or we can make transgenics or block the degradation and induce the ceramide levels and ask the sufficiency question. These questions of sufficiency are still ongoing, so hopefully I'll have some more stuff to share on that in the not too distant future. But we've taken these two enzymes that are required for ceramide production and we've knocked them out of liver, of adipose tissue, of brown adipose tissue, out of myeloid cells, out of muscle, out of the beta cell, out of the vasculature, and the gut. And we phenotyped most of those models. And so I'm going to share you some information. We've published all the adipose stuff. I'm not going to talk about that, but that's turned out to be a real interesting story that really provided the seed for this idea about how they regulate lipid metabolism. But I'm going to tell you about some manipulations we've done with this enzyme here. This is the fourth enzyme in the pathway, dihydroceramide desaturase 1. There are two isoforms of this. One is present in the gut and the skin almost exclusively, and it actually makes a slight variant of the ceramides called phytoceramides. But this dihydroceramide desaturase 1 is pretty ubiquitous. It inserts this, the only modification is this double bond in the sphingosine backbone. The levels of these guys, these dihydroceramides, are very, very low in cells. They tend to be a good marker of disease because they're a good marker of passage of carbons through the pathway. So oftentimes they correlate very strongly, but we think they're inactive, and I'll show you that. But we think this double bond, which is essential for ceramide packing in the membrane, is really important. So we made a mouse, and if you knock it out germline, the animal does get quite sick. So it has a shaking phenotype, seems to have some neuronal problems. A lot of them die in utero. We decided to knock it out in the adult, assuming that eventually some problems would happen, but it would give us a chance to sort of explore and have some tissues and some cells. We were shocked in that when there were no problems with the mouse. We've taken it out now 11 months of age. The animal is incredibly healthy. As a matter of fact, it shows improved metabolic performance even on a chow diet at 11 months of age. The only thing we've seen is that its hair gets a little bit lighter. So it's got brown hair instead of black hair. So it's a superb mouse. We took that mouse, and this is just showing the relative knockout. So we get a nice knockout in a whole bunch of tissues, really good in the liver, really good in the kidney, good in adipose tissue and muscle. And you can measure the effectiveness of this knockout in terms of by looking at the lipidomics, because these dihydroceramides, which are normally really low in abundance, actually go up quite a bit. So you can measure the ratio of ceramides to dihydroceramides, and normally there are a lot more ceramides than dihydroceramides. But if you knock out the gene, you shift that ratio because you get rid of ceramides and you induce the dihydroceramides. So we've got a great marker. This ratio is apparent in the blood. It's a terrific biomarker. This particular set of data are from an OB-OB mouse. So we crossed this mouse, this inducible DES1 knockout, with a leptin-deficient mouse. Again, this is a mouse that gets really fat, eats insatiably, develops sort of an extreme form of prediabetes with impaired glucose tolerance and hyperlipidemia and fatty liver. Now the mouse, the whole body mouse, actually, it didn't get skinnier, it didn't lose weight, but it stopped gaining weight. So we saw a reduction in weight gain, and this was due exclusively to a decrease in fat mass. So it quit putting fat inside its triglyceride stores and showed a little bit of an improvement in perhaps energy expenditure. By comparison, the counterpart, which did not have the gene depleted, this was a tamoxifen-inducible system, but didn't have the transgene, it continued to gain weight and developed the metabolic syndrome. If you looked at the tissues post-euthanasia, the fatty liver that's apparent in the OB-OB mouse was gone. The liver was completely free of lipid droplets. The adipose tissue showed an improved morphology, smaller, more metabolically active lipid droplets. Adipocyte size was down. This is actually looking at the droplet within the liver, and it was also down. If you look at glucose tolerance, there was an improvement in glucose tolerance. There was an improvement in insulin sensitivity as measured by an insulin tolerance test. So this one, we inject glucose, look at the clearance of glucose. This animal normally clears glucose very poorly, but if you knock out the gene, it clears glucose much more quickly. If you inject insulin and look at the reduction in glucose, you see that it clears glucose more rapidly. There's also a reduction in liver enzymes, AST and ALT, suggesting that we've resolved liver damage. Now that's the whole body, but this is a floxed animal, so we now can go in and we can knock this out in every tissue. And so we've knocked it out in myeloid cells in the gut. Nothing happened, nothing good, nothing bad. So I'll tell you why the gut finding turned out to be really interesting later. But so nothing happened in that system, at least with regards to metabolic disease and our measures of glucose tolerance. But if we knocked it out in either the liver, adipose tissue, or liver and adipose tissue simultaneously, we got a comparable resolution of fatty liver and an improvement in glucose tolerance. So to start with the liver, here you see again the lipid droplet that accumulates in the OB-OB mouse in the liver. If you knock out DES1 in the liver, you get clearance of that fat from the liver, despite the fact that these animals, unlike the other ones, they show no difference in weight at all. So these are still really fat mice, equally fat, but none of that fat is being stored in the liver. Adipose tissue, you also got clearance from the liver, but not as much as when you knocked it out of the liver directly. If you knocked it out in both tissues, they looked the same as the liver knockout. The same thing happens in terms of glucose. Fasted and fed glucose were down, whether we knocked it out in adipose tissue, liver, or adipose tissue and liver. Serum free fatty acid levels were also down. So this was exciting to us. It meant this single double bond in ceramides, if you got rid of that double bond in either adipose tissue or the liver or in the whole body, you could have a mouse that was super fat but metabolically healthy. So the question became why, and we had started this whole journey down ceramides because we had shown that ceramides inhibited AKT, an insulin signaling intermediate that was required for glucose transport. But we were very curious about this effect on lipids in the liver. Why would ceramides be affecting lipid deposition? Now, if you improve insulin sensitivity, you should induce triglyceride synthesis, and it's one of the things that's plagued the field is insulin resistance is selective. It's selective for the glucose effects, but actually the insulin effects on lipids seem to go unchecked. And we think this explains it, because we think ceramides actually, as part of their biology, their evolutionary conserved role, they drive selective insulin resistance. They impair glucose utilization and drive lipid synthesis. And these are the data for that. So when fatty acids come in a cell, Jim Hamilton at BU has shown they diffuse through membranes just fine. They don't need a transporter. They actually flow through very, very rapidly, not surprisingly. There are translocases that exist, but those probably exist to facilitate their esterification. And so our idea is that free fatty acids actually in excess in the membrane will lead to destabilization in the membrane in a way that's quite harmful. So we think one of the things that ceramides do is they form detergent-resistant membranes. Now this has been known for years. If you look at, if you purify rafts or caveoli or something, they're loaded with ceramides, and you purify them because you look for detergent resistance. That's the name of the fraction. So one thing we know is that they are a key component of that. So that's been known for years. One of the things that's not been known is that they actually facilitate the insertion or stabilization of CD36 of the plasma membrane. They stimulate the movement of it into the membrane to facilitate lipid esterification. So when we took out primary hepatocytes from the DES1 knockouts, they actually showed a dramatically decreased rate of lipid uptake. So this is a study that Trevor, who's in the audience, did, where we added a labeled fatty acids, and you see fatty acids go into the cell. If you add insulin, they go in a little faster. The DES1 knockout doesn't accumulate lipid to the same extent. And this is just a quantification of that same thing. Control, insulin stimulated. If you knock out the gene, they don't take up fat. And we think that's driven by this ability of ceramides, but not dihydroceramides, to move this enzyme CD36 away from out of the ER and towards the plasma membrane. And they seem to catalyze that release. Now this is consistent with something that Will Holland and Philip Shear had shown in a paper the year before. Another thing they seem to do is they seem to upregulate genes that drive lipid droplet formation and lipid storage. And so we did RNA-seq on those livers, and the only gene that was more dramatically regulated, whether the animal was on chow or a high-fat diet, was SREBP. It actually went down further than DES1. And so the one gene that seemed to be dramatically changed in these experiments was this master regulator of lipid synthesis. We confirmed by qPCR that that gene was indeed down. It was down in the liver knockouts, the adipose knockout, and the liver and adipose knockout. And a bunch of its targets that are involved in lipid droplet formation were also down. So it facilitates lipid esterification. It facilitates its storage. What else does it do? Well, I mentioned that we started this because of its ability to inhibit AKT. AKT is obligate for insulin-stimulated glucose uptake. We mapped the mechanism a number of years ago. It involves two different pathways to inhibit this, to block this activating phosphorylation of AKT. And sure enough, ceramides inhibit AKT. Dihydroceramides that lack the double bond are incapable of doing that. If you knock out the gene, you get hyperactivation of AKT in the liver. So you have an improvement in insulin signaling through AKT in the absence of these ceramides. And ceramides themselves will block the utilization of glucose. Now, the adipose tissue knockouts also had an effect. And so one of the things that was interesting, we got to thinking about this idea, well, if their job, if ceramides are working to lower free fatty acid levels to protect the cell from this fatty acid toxicity, might they affect lipolysis? Might they affect the release of free fatty acids and suppress that? And sure enough, we looked at that, and ceramides, but not dihydroceramides, block the phosphorylation of hormone-sensitive lipase to block the release of fatty acids. And we mapped this mechanism too, and it's again through a ceramide effector, protein phosphatase 2A, that seems to get activated at these microdomains. And then lastly, they decrease mitochondrial efficiency. And we'd shown this in a number of papers, and we showed it again here. If you do manipulations, either adding Ariosin to lower ceramides, you add back ceramide, you add back drugs that block ceramide modification, you can change the cellular pool of ceramides. And then if you look at mitochondrial activity, it's almost an inverse image. if you have low ceramides, you increase mitochondrial efficiency. If you lower it, you decrease or if you increase it, you lower mitochondrial efficiency. Now, I say this confused me, so I went to one of my friends, Jared Rudder, who's a mitochondrial expert. He goes, oh no, that makes perfect sense, because what's limiting in your ability to burn fat is the size of the gradient that gets produced across the inner mitochondrial membrane. If you decrease mitochondrial efficiency, you can actually burn more fat, and that's a way of increasing the flow of fatty acids through. And sure enough, if we knock out the desaturase, we get increased mitochondrial efficiency. If this is true, then you would predict that conditions, biological conditions, where you needed to release free fatty acids would shut down ceramide synthesis. And so the first thing we thought of was if you're running from a bear, fight or flight response, classic, you would want to shut down ceramides because you would want to ramp up glucose utilization and free up all your fatty acids. So we used an assay we developed, which is a stable isotope assay, where we had labeled serine, and then we can measure the rate of ceramide production. And in about 5 hours, half the ceramide pool in a cell gets turned over. So turnover of ceramides is very, very quick. Oh, I should say, there's one other piece of evidence for this. One of the enzymes, one of the ceramide synthetases, was recently shown to actually be a transcriptional repressor. It reacts to fatty acids by not only catalyzing formation of the dihydroceramides, which get converted to ceramides, but it then moves into the nucleus to repress lipase expression. So that was another piece of evidence that gave us, it's not relevant in the DES1 knockout, but it helped give us support for this hypothesis. So what happens to beta-adrenergic agonists if you have the stable isotope? This is looking at the rate of production of ceramide using these stable isotopes. You get here in this cell system, this is a primary subcutaneous white adipose tissue, you get a rapid increase in ceramide production that's completely shut down if you do beta-adrenergic agonists. Even though we're flooding this system with palmitate, we're just loading it in there, but it completely shuts it down. Okay, so I've shown you this whole thing happens, and then what we think happens is when ceramides get really high, there's been a whole host of studies showing that they induce apoptosis and fibrosis, and we think this is just the next step to that. That's what protects the organism when the fatty acid levels get so high that it can't protect itself from these other mechanisms, and it prevents the spillover of cellular contents into the extracellular milieu. There's always one exception, right? So we knocked out DES1, and we also knocked out this first gene, SPT, in all these tissues. They all either got better in terms of metabolic health, or they were unchanged, with one exception. And it was when we knocked out SPT, but not DES1, SPT in the gut. And if you knock it out in the gut, the animals die within 4 days. Eli Lilly actually made drugs against SPT, and they failed because they had a gut toxicity. We don't get this if we knock out DES1. And we've actually mapped the mechanism. What happens is, and if you can see it right here on the right, if you deplete SPT within a couple days, you've gotten rid of the stem cells that form at the base of the crypt. So what we think happens is, for a tissue like the gut epithelium, which has to rapidly regenerate, it doesn't respond to ceramides by rigidifying its membrane. It responds to ceramides by making a lot more stem cells. And we mapped the mechanism using organoids, primary organoids. You grow these mini intestines in culture. We showed that it was not ceramide, it was a downstream metabolite, sphingosine 1-phosphate. It's required for growth. So if you deplete SPT, the cells die. If you add back sphingosine 1-phosphate, the cells live. And we think this is required for stemness. We actually think this is one of the things that may link high-fat diets to increase susceptibility for colon cancer, because you disproportionately activate replication of the stem cell pool. So that's the exception. That's not affected by the DES1 because there's a second isoform in the gut, and also because DES1, the dihydroceramides, can actually be converted to the dihydrosphingosine 1-phosphate, which is still active. Okay, so the last thing I want to tell you about is how. And so, the who, what, where, when, why. We did the journalism acronym, but then they all say sometimes you also need to answer the question how. And our question there is, how do we exploit this to actually develop a therapy for these people that are calling and say my ceramides are high? So if our favorite drug target is the desaturase that I just talked about, The dihydroceramides don't do a lot of the bad stuff that ceramides do. There are a couple other reasons why we think this might be good. One is there's a drug that's been in clinical trials for a long time. It's called fidretinide. It's a retinol-containing drug that has relatively few side effects, all related to this retinoid domain that has nothing to do with ceramides. It causes a reversible night blindness. But the retinoid has nothing to do with its desaturase ability. This has been in up to 2,000 patients for up to 5 years. So there's a whole body of literature to suggest that maybe targeting this enzyme can be done in a safe way. The other thing that's great about it is we've got this great biomarker. We can give the drug and we can see the appearance of these dihydroceramides that lack the double bond. So you can actually monitor target engagement when you add this compound. I'll show you that in a second. Lastly, Joanne Curran and John Blangero have found an inactivating SNP. It's a rare variant in a Hispanic population. It's present in 2 to 3% of these late generation Hispanics. These people have very high dihydroceramides, very low ceramides. They're heterozygous for the gene. And they have decreased 2-hour glucose, decreased cholesterol esters, decreased diabetes risk score, and decreased major adverse cardiac events. So we started a company with a couple of drug development guys out of San Francisco, Jeremy Blitzer and Steve Hallis, and Don Romero is our medicinal chemist. We made a whole suite of DES1 inhibitors. These drugs have good drug-like characteristics. They have good IC50s against, if you use that stable isotope assay. You can monitor the accumulation of dihydroceramide. You can measure the pharmacodynamics by looking at the dihydroceramide ratios. Here in green, this is the plasma dihydroceramide. In red is the liver. And in black is the drug concentration. So the ratio of these dihydroceramide biomarkers completely track. And the drug works. So within 7 days in an OB mouse, we clear fat from the liver by inhibiting this desaturase with CNT2130. There's decreased steatosis. There's decreased plasma triglyceride. About a week later, we start to see improvements in glucose tolerance. Within 7 days, we see a decrease in fasting glucose. And an improvement in insulin sensitivity is measured by HOMA-IR. So, about a year ago, I wrote this article comparing sort of the status of the ceramide literature with the cholesterol literature. I think there are a lot of parallels. I think that there may be a few different therapeutic ways that we could lower ceramides, which may be particularly effective in combination with strategies that lower LDL cholesterol. So this work was done by, this is our group, Will Holland and I. Trevor Tippett did the whole body DES1 knockouts. Trevor's here. Baggy Tracy did some of the liver knockouts. Liping Wong did the drug studies. And I'm happy to answer any questions. Thank you.